I'm going to tell you about some work we've been doing over the years on developing techniques, clinical imaging techniques for tracking cell therapeutic products in vivo. And so today I'm going to focus on MR approaches. And this is work that we've been doing over a number of years. Aspects of this work have been licensed to a company called Celsense, based in Pittsburgh, Pennsylvania. And I'll talk a little bit about Celsense at the end. I have some disclosures here. So what we're trying to address is an urgent clinical need that's really accelerated and intensified over the last 5 or 10 years, which is the area of cell therapy. We're trying to tackle the problem of tracking cells in vivo after they're infused into the patient. And the rationale for this is to accelerate preclinical development and evaluation to look at different candidates. Often in the laboratory, a cell biologist defines a cell by its phenotypic markers on cell surface. They maybe run flow cytometry to identify a panel of markers that are relevant, identifying the cell's identity. But what's the impact of those cell markers in vivo? How does it affect trafficking of organs and tissues in vivo? And so that's something that we're trying to bridge the gap and develop in vivo cytometry approaches, I call them, to really understand what the impact of those cell markers are. And this allows you to screen drug cell therapy candidates and evaluate their efficacy in terms of biodistribution. Also in the clinical realm, the ability to track cells is important to optimize cell delivery, to understand the dosing, to get an estimate of the initial biodistribution of the cell product. With this information, you can derive a surrogate biomarker, which I'll talk about today. Another important parameter to try to assess is cell survival. So if you're putting in 1 or 10 billion cells in vivo, you'd like to know how many of those that cell product survives, what fraction survives. With that information, you also help understand the modes of action and rely less on having magic cells, but understanding more the mechanistic aspects of cells as used for treatment. With this information, you can also get retrospective analysis for responders versus non-responders. So if a patient fails to respond to a cell therapy, the oncologist, for example, in their mind's eye, thinks that the cell trafficking was important, but they have no way to see that. And with that information, if you could see the cell trafficking and the survival of the cell product, that will help you derive more cost-effective and smaller and more statistically significant trials if you can bin out responders versus non-responders with respect to cell trafficking. We also believe that this improves safety surveillance. And we believe that eventually there'll be a potentially we'll believe that there's an FDA mandate for having some form of cell tracking integrated in the cell therapy trials. So the approach we're taking is a little bit novel. We've been developing this over a number of years. We're using perfluorocarbon molecules formulated as emulsions or colloidal suspensions. Perfluorocarbons don't integrate into cell membranes, nor are they water soluble, so we have to formulate them as small droplets, nanoemulsions we call them. And these have been used in biomedicine in the context of artificial blood substitutes for many decades. So the FDA is well familiar with the safety profile of these types of molecules. We like molecules that have a chemical end groups that are amenable to modification, and we can make dual mode agents, for example, putting a bright fluorescent dye, for example, linear perfluoro polyethers. So this is an overview of the technology, in vivo cytometry, where we're looking at cells in vivo and counting them. You take cells from the patient or allogenic cells or cell lines, you culture them as part of the manufacturing step. You then add the perfluorocarbon emulsion as another factor in, and you label the cells in culture. The cells take up the nanoemulsion droplets, generally trafficking them to low pH lysosomal vesicles, where they remain stably labeled as long as they remain viable. We then assay the amount of label that's taken up per cell in a batch. We derive the 19F per cell, the fluorine per cell, using NMR techniques. We then infuse the cells into the patient by any route, and then we do an MRI scan. And so we use the external reference along the patient torso. We scan the patient using an MRI, and clinical machines can be adapted to do fluorine-19. We take a proton scan, conventional MRI. The pseudo color is the fluorine scan. We also have the reference. And then we have software that we've developed that does ROI analysis of the reference, the hot spot of where the cells accumulate, and the noise for the statistical significance. We amalgamate those three numbers to get something which is called the apparent cell number, which is a surrogate biomarker for cell delivery. We also can do this using NMR cytometry. So in preclinical studies, we can take the labeled cells, inject them into test mice, for example, bearing a tumor, and then we let the cells distribute as they would. We necropsy panels of intact organs, and we do intact organ analysis using NMR, where we're counting the cells in each tissue sample of the panel, and then we can then plot the apparent cells per tissue and the lesion. And this is a high throughput, and so this eliminates histology or biochemical methods for looking at cells and counting cells. The time per sample is about 10 minutes per sample. So it allows you to look at many different cell candidates, for example, in a high throughput fashion. The commercial version of this material is called CS1000. It's manufactured and sold by Celsense. It labels any cell type, leukocytes of the various flavors, stem cell and progenitor cells. Any cell type we've tried to label, we've been successful. Here's an early example of the labeled dendritic cell. This is microscopy showing the intracellular distribution of the label. It's perfluorocarbon droplets. You can see a little hyper-intense ovoids of signal inside the cell seen in the EM. And so large numbers of these droplets accumulate inside the cell. Only viable cells take it up. The cell remains viable. It will stay labeled. If the cell dies, the material is dispersed and is generally cleared by the RES system, and ultimately through lung exhalation is the clearance pathway through the body. What you can do then is take a fluorine proton scan and then look at the labeled cells in the anatomical context, where the conventional MRI looks at the background signal, the background anatomy, and the pseudo-color is the cell distribution. What's nice about this technology over prior art is that you can look at the biodistribution of the cells, and you can just look at one MRI scan. You don't need two before and after injection if you're using metal ion agents. And the signal that you get from the fluorine is irrespective of the background tissue that it resides in. So it's an unbiased visualization of cells. Here's another example in T cells where we took T cells, inject them in an inflammation model in mouse where we put ovalbumin and IFA into the quadricep muscle, and then we inject cells systemically, and then you get accumulation in the inguinal nodes. This cell label persists, so even out to 3 weeks or if longer, you can still see the presence of the cells. Recently, we've been looking at adaptive cell therapy, of course. We developed in a CAR, for example, data in a CAR T cell model with a CAR for EGFR variant 3. It's a glioma model. We can look at the cell biodistribution of the infused CAR T cells. And so this is a log plot, so there's a lot of dynamic range. This is done with NMR cytometry. The tissue panels on the x-axis. And you see the biodistribution of cells. You see, for example, the tumors accumulate CAR T cells in larger numbers compared to untransduced T cells, and those cells persist for longer. The liver signal represents the dead cell fraction because the material is cleared by the RES, particularly the Kupffer cells of the liver. So by measuring the liver signal, you can get a good estimate of the dead cell fraction of your cell therapy product. You also see cells in other tissues as you'd expect. The tail is shown here because it's a tail vein injection in a mouse, and the tech sometimes has a little misinjection and gets some cells in a sub-Q space, so you could actually back that out of the effective dose to the animal. We also make dual mode versions of these agents for preclinical use. And so after the MRI or the NMR, we can do fluorescence imaging. This is a really sophisticated type of technique. It's cryo-fluorescence tomography, where it's whole animal sectioning with a fluorescent camera. And you can see the biodistribution of the agent. And the panel on the left shows the Celsense agent. The tumor cells are in green with expressing GFP. Autofluorescence is in the gut. And then you can look at the tumor microenvironment of cells and the tumor cells. It's segmented out on the right. We've put this into the clinic. This is regulated by a drug master file. It's held by Celsense. Our first IND was at the University of Pittsburgh. We looked at colorectal cancer patients tracking an autologous dendritic cell vaccine. And what we do is we work in the manufacturing where we do the labeling. So this is all done in the GMP lab. And we add PFC at 5 days into the manufacturing when they're also acting adding activation factors and doing antigen loading. 2 days later, you can do cryopreservation. The cells stay labeled, do release testing, fused into the patient. We look at very carefully the phenotype and function of cells that are labeled and compare it to unlabeled cells. And this is patient data. We measure the labeling, which is shown in the upper left. We look at the fluorine per cell for each patient. We look at the viability of the cells, and then we look at the release markers that are used for normally releasing these dendritic cell vaccines, compare labeled to unlabeled to make sure that it meets the release criteria. This is MRI data in a patient's. The middle panels show proton fluorine MRI scans with the injection sites. These are intradermal injections. And the graph on the right shows the quantification in vivo cytometry of the injection site. And about 24 hours after injection, half the cells are gone, so they're probably, a lot of them died, and some have migrated into the lymphatics. We also have several clinical trials that are underway at UCSD, so a place down the street here, using tumor-infiltrating lymphocytes in head and neck cancers, and we have RO1 support to look at CS1000 in head and neck cancers, quantitate TIL homing to sites of metastases, but also quantitate apparent dead cell fraction by looking at the liver signal. We've also been developing new products for sensitive imaging of cells in vivo, and we've been working with Celsense and Takeda Pharmaceuticals to second-generation PFC agents, so-called FETRIS, with greatly enhanced sensitivity over the first generation. And we're going to do the same patient population so we can compare A versus B cell labeling methods. We've also, in my academic lab at UCSD, we're working on next-generation PFC cell tracking agents. So I mentioned the PFC that we're using in the TIL trial, and that is now, we've now constructed a drug master file for this new agent. And this is enabled by novel metal chelates that we solubilize in the fluoresce phase that greatly accelerates the data acquisition time of the fluorine-19 scans by, can be dramatic amounts, which allows you to do more signal averaging in a fixed scan time that you have limited time with the patient in the scanner. So this is working great. We've also been developing peptide-targeted cell uptake to enhance the amount of labeling that gets into cells and to speed up the process. And so this has allowed us to achieve 8-fold improvements in sensitivity. And so we're very excited about these developments. We're also looking at positron emission tomography PET probes based on the same technologies that early work looks extremely promising. Celsense, the company, generally delivers imaging tools for discovery and preclinical market, translational cell therapy, and diagnostic medicine. They have a catalog of perfluorocarbon agents, the CS1000 agent that is suitable for clinical trial use. We also have a product line called VSense, which is for inflammation macrophage detection in a broad range of inflammatory diseases. We also have developed a software product called Voxel Tracker to amalgamate the fluorine proton image and to quantitate the amount of cells and regions of interest and the uncertainty associated with that number. They also provide services in cell labeling, protocol development, preclinical studies, and IND development. So to summarize, key features of cell tracking: high selectivity for labeled cells, no background from unlabeled cells. The fluorine is not normally present in the body, so any signal that you see is what you put in. So there's really exquisite specificity for the cell labeling product. You can do whole body screening. Conventional proton images place the hot cells in the anatomical context. You can do cell quantification, which is really a major advance over prior art and MRI. Very low toxicity associated with these probes and is amenable to clinical use. Thank you very much. Yes, question? Pardon me? The limit? Yeah, so it's dependent on cell size. So small cells you can label with less media than larger cells. So for the dendritic cells, we benchmark the sensitivity about 10 to the 5th cells per voxel, which is the smallest volume element of the image. So you're not seeing single cells, you're thinking thousands or tens of thousands, maybe 100,000 cells. In the NMR cytometry, the sensitivity is much higher. It's about 1,000 cells per sample.